Prior to the Hershey and Chase bacteriophage experiment, most scientists believed proteins were the material that transmitted genetic information from the parent to their offspring. In 1952, Alfred Hershey and Martha Chase confirmed that DNA is the material that transmits genetic material. The experiments conducted by Hershey and Chase involved labeling the different phages. For the experiment, the scientists decided to use bacteriophages to infect the bacteria cells. Remember that bacteriophages are a type of virus. A bacteriophage has two main parts, the DNA and the protein. The protein was labeled as 35S and the DNA was labeled as 32P. Sulfur is used and taken up to the protein phage, and the phosphorus is taken up in the DNA. Now that both are split, you have two separate phages. E. coli was mixed with the labeled 35S and 32P phages. Scientists blended them together to shear off the protein coats and determine if the radioactive material was on the outside or the inside of the bacteria cell. Once blending was complete, the liquids were placed into a centrifuge and spun down. This resulted in a supernatant and a pellet, also known as the protein and DNA. 32P demonstrated that DNA was in the bacteria, while 35S demonstrated that protein did not enter the bacteria cell. This shows that the sulfur or protein was in the supernatant, and that the protein or DNA was in the bacteria cells, which is the pellet. Hershey and Chase detected that the radioactive material was in the pellet, and that led them to confirm their beliefs that DNA is indeed the genetic information in bacteriophages.